on in my lab is trying to understand then the mechanisms that are underlying vulnerability for alcohol abuse. And specifically, we focus on understanding the mechanisms that drive the punishment-resistant alcohol drinking. How is that alcohol persists despite adverse consequences? And we focus on this subject as one point that we think is very important for defining the compulsive aspect of addiction. So I want to, first, because I know this is a very international crowd, I want to actually place in the map where we are. And my lab is part of the National Institutes of Health, which is located in Bethesda, Maryland, within the United States. And we are, like most people around the world, pre-quarantine these days. So this is a recent picture from the lab meeting. So where some of the folks are able to be in the lab. And so we take turns going in, and the rest of us do the work from home. But as the vaccination is coming around, so we're hoping that we can get more and more in the lab. And I imagine that that's the similar trend around the world. I just wanted to bring that up. So we are in the lab very interested in understanding the vulnerability for alcohol abuse. And I always like to bring up these statistics numbers. So in the United States, approximately 1/3 of the adult population consumes alcohol. That's approximately 140 million people. However, a subset of these individuals only engage on binge drinking, and only 12% of those that consume alcohol develop this heavy alcohol drinking that is associated with abuse and disorders. So in average, only 1 out of 8 individuals in the US that consume alcohol displays alcohol use disorders. So the questions that we are interested in understanding in the lab. So we are very interested in understanding what drives that vulnerability. But I want to emphasize how substance use disorders is actually characterized by a wide range of features. And I think many of these have been discussed throughout the course of these seminars. And as defined by the DSM-5, these phenotypes and expressions vary from cravings, withdrawal symptoms, development of tolerance, choice over other rewards, that I think there had been a lot of talk from Serge Ahmed's presentation here. And individuals that suffer also from substance use disorders develop a high motivation to obtain the drug. Basically, they pay a huge price. This is monetary price, but many times also it's just the amount of time and resources they spend on seeking and using the drug and recovering from drug use. And more importantly, 6 and 7, an inability to stop the consumption, even despite the individual saying that they really want to stop consuming the drug, that or planning, actually, for example, going to the bar and planning to just have one drink and then end up having 10. Those are the specific features that our research has focused on more the compulsivity aspect and inability to stop or control intake. And so the continuing of the use despite punishment is something that we try to model in the lab, and I'm going to show you examples of how we do that, and as ways of measuring that inability to stop or control the intake. So the lab has focused on the last 5 to 7 topics, and we are, in addition, interested to understand then what makes some individuals more vulnerable to develop these substance use disorders? What are the factors that contribute to the vulnerability? And how is that the vulnerability happens? So basically, we're interested in not just identifying genes or circumstances, but actually trying to understand how is that this is working in the brain to drive these behaviors. And the goals then are to identify the molecules and the circuits that are driving the reinforcement properties of alcohol and other drugs, and also understand how the transition happens from this hedonic to compulsive taking, with the goal that we will develop treatment for improved treatments for substance use disorders. So now, I don't know if this just happened to me, but when it might be something that also is shared by other viewers, that when faced with those statistics from the human population, one simple guess is that assume, well, there are, of course, genetic reasons why some individuals are more vulnerable. We know that within the human population, there is a significant diversity in our gene pools, and so that could be an easy explanation. But I'm going to try to make the point that actually it's a lot more complicated than that. And in large part, the evidence is coming from our own animal research, where we observe that, for example, within inbred C57 black cis J mice, when we give them access to alcohol in the home cage, and this is, they voluntarily will drink alcohol, or some of them will, as you will see, and we give them 20% alcohol in tap water, no sucrose, no saccharin, no, it's just 20% alcohol in tap water. And to give you an idea, red wine is 15% alcohol, vodka is 40%, so we're giving them something in between in tap water. What you can observe is that even on that first session, there is a wide range of variability on how much the animals will consume in grams kilogram. And just estimate 4 grams kilogram is what in 4 hours is what will in average reach intoxication levels, 80 milligram deciliters on the blood. So some animals are consuming a lot, many drinks that are highly likely to get intoxications. Other ones are not. Now, when we look over the days, in addition, we observe that there is variability across the days. So some animals that, for example, this animal that didn't drink much on the first day now goes on to drink a lot, and so on. And we observe, like in humans, okay, so first, individual variability on the drinking, even on the first day. We also observe variability, daily variability. And I wanted to point out that based on our observations, when we give animals access to alcohol on repetitive consecutive days, that we observe that animals that might drink high levels for a couple of days, but if they do so, they are likely to actually don't drink on the 3rd and 4th days. Like they kind of seem to take breaks. So we're even adapting our drinking paradigms, and we're giving access to animals every other day because this seems to better synchronize them on their drinking. So that's another observation that comes from that. The other interesting observation I wanted to make that pointing out to differences or factors that affect how much animals consume alcohol is that we also observe, like many other labs across the world that have been doing these studies, that there are sex differences on drinking behavior and that female mice drink more alcohol than males. This is an example from other set of data on how when you look across now, average across the days, sessions, in average, male mice drink less than females. And that is a quite consistent thing that I think also is observed in rats. So what happens when we get these animals now in an operant paradigm? And this is a picture of the front panel of the operant chamber, where there are 2 levers, an active and an inactive lever. When the animal presses the active lever, gets access to a zipper that otherwise is covered with a door. And very importantly, for these measurements, we are measuring not just the number of lever presses, but also we are measuring leaks, because there is a leakometer connected to the zipper on the floor, so we can get an idea how much when the animal gets contact with the zipper. But more importantly, we're also drinking the volume that the animals consume. And throughout the point the lecture, I'm going to be discussing some of these issues as well. So we can train the animals on self-administration, first on FR1 and then FR3, and get different measurements of their motivation on a breakpoint session. For example, asking the animals to now press more and more times to gain access to to the zipper. There are also sessions in which the alcohol is paired with quinine, a bitter testing solution that actually it's quite aversive. And so animals, for the most part, will then restrain from drinking during those sessions with reduced drinking. And also sessions in which the alcohol drinking is paired with punishment, with a food shock as another way of measuring punishment. So those are some of the things I've been doing. So what we observed that even within these C57 black cis-J mice that are genetically identical, or 99.5 identical, we observed that not all animals engage on the self-administration task. So we can separate two groups, 60% of the animals in this experiment, but it seems to be quite consistent for this strain, will show considerable intake and and then 40% of them will actually don't consume under the operant. Interestingly, look what happens to the lever presses. Even the animals that do not drink alcohol, based on what the volume that they have consumed, they will engage in some degree of lever pressing. And I think this is an important distinction that I will keep making up as the progress, as the session comes on, as to bring up the issue how we really need to measure consumption, not just lever presses, to distinguish on the behavior. So unsupervised cluster analysis of these animals groups, for example, show that you can discriminate within the drinkers, that now are 25 of those 40 animals we started, 3 groups. And this was input the data of the intake, how much animals were drinking throughout the sessions, and how they decreased their drinking when paired with quinine, and the % drop in the intake when it was paired with a food shock. And this cluster analysis, so here is the cluster analysis data, shows that there is pretty good separation across these groups on the analysis groups, and also shows the sex distribution of these 3 groups. And you can see for the most part, the group 2 and 3 have similar amounts of males and females, but group 1 has a higher percentage of males, which might give you some clues of how these animals then acquire the behavior when we sort them out. So here is how their intake is when we sort animals based on these groups. And what you can observe is that the group 1 actually shows very low level of drinking, but the other 2 groups eventually reach the same levels. However, the red group, that here gives you a hint, it calls resistant, shows much more escalation in the drinking consumption. One little more example of what happens and why it's important to measure consumption is that if you were to try to separate these animals based on their lever pressing behavior, you will notice that this actually is much harder to do. For example, the red group here that you're going to see differs on the punishment resistance, actually does not behave very differently on the lever presses. However, is consuming a lot more than the group 1, that is the blue. So what this means is that animals can actually lever press to get access to the zipper, but that doesn't mean that they're actually consuming or drinking. But when we measure that, we can tell the distinction. So these 2 groups actually differ on their response to punishment. And so this is what we can observe here, that the red group continues. So if you measure how much they consume before the punishment sessions and during the punishment sessions, there is no drop. While the group 2, that green group, that also drank equal high amounts, now significantly drop the intake during the punishment session. So we are referring to this group, the green group, as the punishment sensitive and the punishment resistant. And again, this is, if you were to look at level presses, you wouldn't be able to notice this. So again, it's very important that we, especially when we look at alcohol, that we look at consumption. I want to point out that the three groups did not differ on their breakpoints or their motivation, as we'll see, or effort to seek alcohol. Neither they differ on how much they drink on the sessions before they went on to the operant. So this really points out that this behavior on the response to punishment is quite selective, and it can only be discriminated when we look at the operant paradigm. We cannot tell this by just looking at voluntarily alcohol drinking or differences on how much they will drink before voluntarily. So all this to say that clearly this data is showing to us that the factors that determine the vulnerability for developing this compulsive alcohol drinking are more than just the genes. And our current hypothesis is that what drives the vulnerability is actually the state of the brain circuitry that is controlling these behaviors. And the reason why this matters, that the vulnerability is determined by the connectivity, we think it's very important because it allows then for conditions in which then genes and multiple genes, not just one gene being the factor, multiple genes combined with environmental factors, combined with internal state factors, can interact to generate a vulnerable circuitry. And this could happen, so this is a kind of our working hypothesis, that this can happen at some point early in development and be fixed, but also if it is driven or affected by internal states, like stress, or any other internal stress, it's possible that there are situations in which the circuitry becomes even more vulnerable or becomes vulnerable but is protected under other internal states. So this is our working hypothesis. And with that, I'm going to now go on to show you examples of two genes or two molecules that we think are very important in determining the state of that circuitry and that can generate in some ways this vulnerable circuitry. But I want to clarify that I don't think these are the only factors that are playing a role, that is very likely that a lot of other genes can also be playing a role. And also that the studies we're doing have focused on the striatal, which is one of the nuclei within the circuitry, but I am sure that there are very similar mechanisms that are happening in other brain regions, such as the prefrontal cortex and other cortical regions, the amygdala, and lots of other places, the midbrain. All the other regions that are part of the circuitry are probably undergoing very similar changes that could contribute to the vulnerability. So those clarifications then made, I will then move on to tell you the story of some of our work that it starts off with some very interesting observations that were done in humans. And actually, this is the work of two really amazing women in neuroscience. One is Andrea King and Harrietta DeWitt, who had been looking for many decades, I will say now, at these vulnerability factors or associations and alcohol abuse in humans. So it turns out that in humans, a stronger alcohol stimulation is associated with the likelihood of alcohol abuse later in life. And so this is an example here, individuals that were tested eventually later positive for alcohol use disorders. And in the initial testing, they showed that when they were administered alcohol, a subjective, enhanced subjective stimulant response to alcohol compared to those individuals that actually turned out not to show alcohol use disorders. And so what they show in this study, follow-up study that just came out ahead of publication, is that this initial observation that was done on the Chicago Social Drinking Project was follow-up over 10 years and remain true even after 10 years. And they even see hints of some sensitized effects of this stimulant response to alcohol. I want to point out that parallel to this, alcohol produces also strong sedative effects that they tend to develop later. You can see how here the stimulant effects are fast and transient. The sedative effects seem to develop slower with time, and they last longer. And what we can see here is that the relationship is inverse. Actually, individuals that went on to have alcohol use disorders actually show a lower subjective sedative response. And I want to point out that, so, and to be honest, this was, I think, in the initial observation. People observed that individuals that were not sensitive to the sedative effects were more likely to become alcoholics. And later, they understood that those individuals actually were more sensitive to the stimulant effects. And the reason why I think this is very interesting is because I know individuals will say that we all drink to relax. I don't know how people will describe it, but that's how. I think I always like to say, like, if you walk into a bar, people are not really relaxed. So I will argue that the reinforcing properties of alcohol mapped a lot more with like the stimulation or the euphoric effects of ethanol than actually the sedative effects. It's also possible that the effects of alcohol are sensed more as a disinhibition, which might feel like a relaxation, but is not really an anesthetized state. So this is what has been observed in humans. And I want to show you that in C57 Black 6, we can also measure stimulant effects of alcohol. And so we can inject animals with alcohol, and we can observe, especially during the 5 minutes, they show a transient increase in their locomotor activity. And just like in humans, there is a large degree of individual variability on the how strong these stimulant effects are, compared to how they respond at saline. That is 100% of the response. That would be just like saline. Some animals show profound stimulation, like almost double, but other ones showed very similar. So there is, even within inbred C57 mice, a widespread individual variability in these stimulant effects. And we were able to show that the degree of this stimulation by creating a score where 0 corresponds to this 100%, there is a positive correlation on how much the animals drink and the levels of intoxication they achieve during those drinking sessions. So we can also, so the more stimulation, the more alcohol drinking in the rodents. And also if we measure now the sedative and anesthetic effects of alcohol, we can also observe, which Miriam Bokarshli did in the lab by using the loss of writing reflex. We can observe also individual variability within C57 mice, and now an inverse correlation with the blood ethanol concentrations. So how much alcohol were found in blood after a drinking session. So again, less sedation like will correspond here, more drinking, more sedation, less drinking. So it seems like this is very similar to what is observed and is something conserved throughout the rodents. And so it turns out that then we can observe this as good evidence that a heightened alcohol stimulation in rodents also seems to lead to alcohol, or it could lead to heavy alcohol drinking. Another vulnerability observed in humans, and this is actually the work mainly of Nora Volkov, but also other groups and other very influential female scientists in the field. Her group showed that low levels of D2 receptor availability in the striatum of humans was associated with alcohol abuse, and even animals, even individuals that were in detoxification, they had been abstinence from alcohol use for several months, still displayed low levels of D2 receptor availability. So this leading to that maybe this was either a persistent reduction or something that preceded the alcohol abuse. So what I'm going to show you evidence from our animal models that there is actually a link between these two vulnerability factors that were observed in humans. And I'm going to show you that actually low levels of striatal D2 receptors then can also lead to a heightened stimulation and a higher preference for alcohol. So this is work that was published last year or 2 years ago by, it's work by Miriam Bokarshli. And what she did is like produce a genetic deletion of D2 receptors specifically in different cell types across the basal ganglia circuitry. And she showed that specific deletion of D2 receptors, or a reduction, I will say, that also of D2 receptors in a striatal spiny neurons was sufficient to increase ethanol stimulation. So these animals, transgenic animals, are called IMSN DRD2 knockouts. They lack D2 receptors only in the indirect pathway spiny neurons. And when injected with alcohol, they display an enhanced locomotor response compared to their leader-made controls that are the D2 flux animals. And you can observe this in this dose response that the control animals showed this stimulation that is approximately 50% from baseline, and they're at 2 grams kilogram. So it's a dose dependent stimulation. And the animals that lack D2 receptors, either in homozygosity or heterozygous mice that show a partial deletion of D2 receptors just in these cells, have an upward shift in the dose response. So even at doses in which wild-type animals won't show a stimulant response, these animals are already showing stimulation. And at the peak of 2 grams kilogram, they also show an enhanced stimulation. Importantly, Miriam went on to show that animals that lack D2 receptors in these cells selectively, they prefer alcohol more. So they have higher preference than the wild types leader-made. So again, low ethanol, low D2 receptors, then in this case shown to be sufficient to induce alcohol stimulation and lead to higher preference. I want to point out this very nice complete study that she did in there. We also generated transgenic animals that lack D2 receptors in other cell types. These are the RD2s, they lack D2 receptors in the dopaminergic neurons, or the SYN DRD2 knockouts, they lack D2 receptors in the cholinergic interneurons within the striatum. So these are receptors bindings that could be contributing to this D2 receptor availability. So she also knocked them down there, and she showed that all these strains of mice, all these transgenic animals showed very similar dose response for stimulation compared to leader-made controls. So no change, but that the potentiation was selective for animals lacking D2 receptors in IMS sense. I think that's really nice. Then in collaboration with Daniel da Silva in the lab, they ran these operant self-administration experiments I kind of showed you early on. So put the animals in the chamber, get them to lever press for alcohol, get access to the zipper. And surprisingly, what we found is that the animals that lack D2 receptors in IMS sense show a much more pronounced escalation of the drinking. And I want to bring this up because it wasn't only until recently that Daniel collected that data that we can understand that this escalation resembles very much the escalation that we observed in that group that was punishment resistant. And and that was selective for this transgenic again, and not the transgenic animals that lack D2 receptors in the dopaminergic neurons, the RD2s. So this pattern was very selective. At this point, early on, we didn't make much of it because we didn't know how to interpret it, but there are higher percentage of escalation. But more importantly, these animals that lack D2 receptors in the IMS sense also are punishment resistant. Saying that when alcohol at the end of this paradigm was combined with quinine, adulterated with quinine, most animals, control animals, most of them reduced their drinking, and so the RD2 knockout animals reduced their drinking, but not the animals lacking D2 in IMS sense. So we think that this data shows that selective reduction of D2 receptors in these striatal neurons can drive the enhanced stimulation response to alcohol, which has been shown in humans to be associated with high alcohol drinking. It can then lead to also enhanced preference for alcohol in the rodents and an escalation in the operant self-administration paradigms, and also resistant to punishment of alcohol drinking. So these are very important. They're small pieces of evidence, but put together very important validation that there is a link between low levels of D2 receptors in the striatum and the enhanced stimulation, and that these two factors through the link lead to the escalation of drinking and some of the phenotypes that are seen in individuals with alcohol use disorders. Now, what is that link? In that study from in Bocarsley et al. 2019, Miriam shows really nice evidence that sensitized D1 receptor response seems to be an important part in this link. And you should go look at that data because it's, I think it's really nice. She has behavioral evidence and also the work of Lauren Dobbs, also previously in these animals, has shown that there is behavioral escalation and other changes that the one receptor response that you should look up. But so the way we summarize this study is that we think that the low levels of D2 receptors are generating this D1 receptor hypersensitivity that is then driving the alcohol stimulation, the escalation, the preference, and the drinking despite outcomes. So there are two main questions that remain. One is, what are the mechanisms that drive this D1 receptor sensitivity? And I think we're still in the loop for them, but I'm going to show you evidence of one gene that could be playing a very important role in this. And the other question that I might leave probably for the discussion session is, how is that low levels of D2 receptors, which we know are expressing one cell type within the striatum, can affect or lead to changes in the D1 receptors that are expressing a different cell type on neighboring cells? How is this mechanism happening? And I think I can show you some evidence of how we think this is happening, and the experiments that we're going to do to continue. So in the next 10 minutes that I have, I want to tell you about this gene, this leucine-rich repeat kinase 2. That is a gene that is a Parkinson-related gene, because it actually, mutations in this gene are the most common form of the familial cases of Parkinson. And this gene is leads to a very complex protein with multiple domains, scaffolding domains, and also kinases and functional domains. And its function is not fully understood, but there are pretty good evidence that LRRK2, as I'm going to refer from now on, can regulate dopamine receptor signaling. And there are evidence that can do those things by two main processes. One, because LRRK2 has an ACAP function that can regulate pKa localization and activation, and pKa is downstream of these G protein coupled receptors that are the dopamine receptors. So downstream signaling through D1 and D2 receptors involves changes in pKa activity, and LRRK2, because it regulates the localization of pKa, can be then implicated in this. And also there's pretty good evidence that LRRK2 can be regulating also the trafficking and the surface expression of dopamine receptors at these medium spiny neurons. So LRRK2 is highly expressed in the striatum. This is a picture from the Allen Brain Atlas, but I'm going to show you data from Daniel's also, RNA-scope data, showing that if we localize LRRK2 puncta, mRNA puncta, with markers for D2 MSNs, here, for example, LRRK2A3, or D1 MSNs, the D1A gene, we can see that actually LRRK2 expression is localized to both groups, and this is like both cell types express varying levels of LRRK2 gene, and from other cell types within the striatum are some of the largest expressors of LRRK2. So what Daniel did is generated cell-specific knockouts for these animals, and I'm going to focus on so we can have the green animal that lacks LRRK2 in D1s, MSNs, or the red animal that lacks LRRK2 in the D2s, and also the full knockout. He shows that then he can get a selective reduction in the striatum that is, of course, partial under both of these animals, because only is one of the subtypes each, but that doesn't, there is no effect in other tissues such as lung, except with a full knockout is here. And so what he observed is that selective deletion of LRRK2 to D1 MSNs indeed will cause changes to the D1 receptor response that were selected for that animal, but not when we remove LRRK2 from the D2 cells. So if we do a dose response for a D1-like agonist, these animals respond more. Some evidence of this D1 hypersensitivity. We also have functional cellular data that there is an sensitized D1 receptor response in these animals. if we measure from firing from the D1 MSNs and their whole cell touch, and the whole cell, recordings, we can then pass current into the cells and measure their firing. There are no changes on the baseline firing of the D1 MSNs in these animals, but in the presence of a D1-like agonist, the animals that lack LRRK2 in D1 cells show a selective increase in the firing on this input-output curve. So in fact, loss of LRRK2 in D1 MSNs can lead to an upregulation of D1 receptor signaling and function. One last piece of evidence that I think this might not be the best example, but we look downstream into the release of GABA from these direct pathway neurons that project to the midbrain. And we can then record a synaptic current on the postsynaptic cells from these cells. And we can measure what happens when we path apply D1 agonists, that there is a slight potentiation on the GABA release, and the potentiation on the GABA release was larger in the animals that lacked LRRK2. So another good piece of evidence from Ayamasui that there is a functional upregulation of D1 receptors in these animals. So we went on to see how these animals respond to alcohol. And what we can see is that, again, the alcohol response stimulation is very transient, but the animals that lack LRRK2 in D1 cells show an enhanced locomotor response to alcohol. And these are at different doses. So there is this upward shift in the alcohol response that is only seen in those that lack LRRK2 in D1 cells, but not in the D2 cells. No changes in basal locomotion. So what Daniel went on to do now is to look at what happens, what is this? Or how the animals drink alcohol, first in a voluntary setting in the home cage. And we see again evidence of this bidirectional modulation on the drinking. So if you remove LRRK2 from D1 cells, they seem to drink a little bit more. The other ones, they drink a little bit less. And he showed that even at different doses of our concentrations of alcohol, 10% and 20%, these animals selectively drink more. Okay, sorry. This is the data from the D2s. So this was selective. And now in the operant paradigm, we can measure now the drinking under operant conditions, and it validated that there is an enhanced consumption in these animals. We can see that actually a higher percentage of animals qualify as drinkers compared to the 60-40. Again, this is a totally different set of group of animals, and we get a 60-40 split. But with D1 LRRK2 knockout animals, the split is pretty shifted. So we think that indeed this gene can be contributing to some vulnerability for alcohol abuse when it's not there. So the function of this gene, interestingly, could be protective for developing these alcohol use disorders. The final piece of evidence from Daniel is that not only the animals drink more, that if you then measure this punishment resistance of the alcohol taken in this operant paradigm, and we can, in this case, he calculated C-scores from this, we see hints. It turns out there is a mild change in the motivation, but more importantly, we see changes in the resistance to punishment with food shock, but also adulteration. So the animals that lack LRRK2 have a higher scores for this, meaning that they are more resistant to the punishment that the littermate controls. So putting all this together, we think that low levels of D2 receptors in the striatum are driving D1 receptor hypersensitivity that also could be achieved by low levels of D1 of LRRK2 activity, for example. So again, there are many factors that can contribute to this D1 receptor hypersensitivity. But once this D1 sensitivity is expressed, this represents a high vulnerability factor that drives more stimulation for alcohol, and we think then more reinforcing properties that leads to the escalation and the preference and the resistance to punishment. And so with that, I think I'm just repeating that alcohol stimulation is positively correlated with preference and drinking, as I showed you, that low levels can heighten the stimulant response, and that there is a link, and that LRRK2 then is a gene that can be playing a very important role in regulating D1 receptor signaling. And again, we think that because when we remove this gene, we generate the hypersensitivity, we think that the function of the gene is under physiological conditions, putting a restraint on D1 receptor signaling, and might have protective factors for developing alcohol use disorders, which will be potentially very relevant for then therapeutics. And this is a picture of the group pre-pandemic times, where we could all get together, and we can't wait to go back to that. Want to point out Daniel Da Silva, who did the work on the LRRK2, Miriam Bocarsley, Aya Matsui, who also worked together. Erin has been contributing a lot to the self-administration paradigms. I need to thank, of course, the funding of NIAAA and NIDA, and also the Center on Compulsive Behaviors. And thank you for your attention.
Wonderful. Thanks, Veronica. That was a really great talk. I'm wondering, while we wait, we'll just give it a second for the questions in the Q&A to pop up if everybody wants to type their questions there. I had a really quick one that was more of a clarification question about the punishment task. So you had this dissociation where the punishment sensitive mice drank much less ethanol but pressed the lever more, or there was less of a difference in the lever pressing. And I'm wondering, when the punishment was delivered, was it with the lever press or with the actual interactions with the lick spout? And what was the probability of getting the shock?
Yeah, it was with the lick, with the lever press. But let me, if I may, go back to share then. So what I wanted to do briefly is, are you referring to the first data set? These guys?
Right.
Okay.
Yeah.
So, and also maybe even the previous one, sorry, here. So I want to point out something first, is that these animals that are the resistant, punishment resistant, in the end, they don't drink differently once they just take longer to reach to that criteria. And we still have to understand why that is happening. One potential interpretation is that they are more sensitive to alcohol. So maybe that early on they just have enough here, and then it's hard to know, or that maybe they're just slow learners. That's also another possibility. For sure, that's how we interpret the data on the animals that have low D2s when they show that escalation. We just thought it was a performance issue and a learning issue. But now that we see this within C57 black cis Jays, where we know that they are not necessarily those factors, we have a different interpretation. So first of all, that's what I wanted to point out, that in the end, they all end up drinking the same. You're referring of how they press differently here?
Right, so maybe that was the clarification, that the punishment sensitive are pressing the lever more, even though the lever press is associated with the shock.
Well, these are not before the shock.
Okay.
And then, okay, with the shock, is this data?
Yeah, I think that was the clarification point. So that's a good answer for me. And I think Anush can answer maybe some of the questions in the Q&A, since we have a couple there.
Excellent. Thank you.
So Vernon Garcia Rivas asked, really nice talk. Are there sex differences in sedation versus euphoria effects of alcohol in rodents and or humans?
Yes, there are. Yes, there are. And I think there is already some published data out there, and I have to now bring up the names. But in our lab, we have evidence that females, as expected, they are less sensitive to the sedative effects as well, and they have they are more sensitive to the stimulant effects. So.
Okay. So David Wenshenker asked this question: is the individual variability in alcohol-related behaviors you see unique to C57 mice, or do other strains show this too? Do other strains have different proportions of mice and different sensitivity/resistant groups?
Yes. Okay. Hi, David. That's nice to know that you were in the audience. And I know, so things that we know for sure is that other strains will drink differently. Some of them seem to have higher preferences than others. I don't know how much there is on the individual variability, but specifically, which was what David was asking, but for sure different strains will drink differently, and I would expect different sensitivities. We haven't looked at the individual variability in other strains.
Okay, so he has another question, which is in Parkinson's disease, the effects of LRRK2 are thought to be mediated at least by expression in microglia. So have you considered role of striatal microglia in LRRK2 in alcohol phenotypes?
Yeah, this is a really great question. Within the striatum, we see that the larger cell types that express LRRK2 are, because doing the RNA scope, we also can measure within non-neuronal types. It seems the largest level is in the medium spiny neurons. Clearly, it's not selective expression to MSNs, so there are other cell types, and microglia is likely to do that. So we haven't looked at the actions in microglia, but I think it would be a very likely, I mean, it would be an interesting thing to follow up after the observations. The full knockout for LRRK2 surprisingly didn't show changes in the drinking. We're still trying to understand how that means, whether they are balancing each other or what. But for sure, those with a full knockout will be lacking in the microglia. For sure, and we didn't see. But again, a selective loss in microglia cells could be playing a role. So eventually, we would like to look at that too.
Okay, and we're just about at the hour, so but maybe we'll have two more quick questions because we have a couple in the chat that we won't get to, but we'll try to squeeze in two more. That's okay.
Yes.
So Christian asks, the punishment-resistant drinkers escalate more but appear to start out drinking less. Could they be overcoming an initial aversion to ethanol that primes them to punishment/alteration resistance later?
Yes, I absolutely think it's true. Good observation. We need to start understanding what's the mechanism that drive that slow start. And that's a very interesting possibility. So we will test that. Thank you.
And Ryan Shepard asks, hi, Veronica, really insightful talk with a lot of interesting data. Two questions. With the correlation of sedation and drinking behavior, have you considered alterations in GABA-AR function and or trafficking that could also be playing a role, particularly in the sedatory mechanism? And
yes.
Yeah.
Yeah.
And
yes.
Two is for LRRK2. Have you examined whether downstream pKa signaling from D1 and D2 or aka P150 is altered using inhibitors such as PKI?
Yes, those are really great questions. And of course, we are looking at how, GABA effects. And I just show you that, for example, the D1 receptor activation that can modulate GABA release is altered in these animals. So we think that, of course, that's probably happening, and it's very important for regulating drinking and the effects of alcohol. So we're diving into now the molecular and synaptic changes. And it's a really great suggestion to look at downstream with PK signaling that we started doing with Western blot, but we will continue. Thank you, Ryan.
Okay, wonderful. So we are right at the hour. So I'd like to thank Dr. Alvarez again for a really stimulating talk. It was really great. And next week at the same time, we will be hearing from Tommaso Patriarchi. So thank you everyone very much and for your participation.
Thank you, Megan, again.